first of all, why don't you just tell me what is the Ingenuity Lab?
Ingenuity Lab is a unique organization that was put together by the by the government of Alberta. They were focused on enabling economic diversification by working in new areas of technology and discovery that mattered. So what they asked for me to do when we helped form Ingenuity Lab was to tackle over the horizon challenges that would ultimately yield to solutions to some of society's greatest problems, and at the same time provide a foundation for technological development that would enable new industries to evolve and expand the economic base of Alberta.
Right on. So how were you sort of the figurehead of this? How did you get involved?
How did I get involved? It's a 10-year story. It began with me being on the external advisory board for the creation of the National Institute for Nanotechnology in Alberta back in 2001, 2002 timeframe. Then I was called on to chair a commission that was being put on by the province of Alberta. It was called the Committee on International Opportunity Assessment. I think that was the formal name of it. So I chaired this international panel, and the focus was how could we use the surplus funds that came from oil revenues at that time, from the royalty revenue checks, to enable more rapid and diverse economic growth. Through a multi-year process, a number of accelerators were identified, accelerators in terms of places where you would invest resources to accelerate technological development in certain areas. And one of the areas that was identified was in nano-enabled materials that have a foundation on biological nature type of platform knowledge. So this became ultimately Ingenuity Lab, where our motto is the power of N, where N stands for nature, nanotechnology, and networks. We take inspiration from the way nature and the world around us works. We use nanotechnology for manipulating matter at the smallest scales to try and emulate the way nature works, and then we incorporate it into systems or networks that are economically translatable to become real technology, not just science projects.
Right. Yeah. Well, it seems to me emulating nature is always a good route to take. It's had even billions of years to develop into what it is today. So it always seems like that's
Well, it may not be the best way to take for our purposes, but it definitely shows a path.
right.
And
Provides
and the details of how you embark on that path may be different, but it definitely illuminates a path forward for solving some of the big challenges. So, our Carbon X Prize program, we have a technology that we've developed that for carbon transformation, and in our technology, we figured out how to take light and convert it into chemical energy, very much the way plants do, but it's not quite photosynthesis. It's something that we've engineered, but we use components from living systems, and then we use that chemical energy to power a metabolism like you would find in plants, to take that CO2 and make complex carbon molecules with it that you could ultimately sell. So we figured out how to take light, convert it into chemical energy, then use that chemical energy to transform CO2 into value-added products, just like a plant would do, except in our case, the products that we want to develop are things that plants don't produce oftentimes, and we're able to do with higher efficiencies because we don't have to maintain the physiological requirements of a living organism.
Right. And just coming back, do you see Alberta and Edmonton in particular as a leader in these sort of advanced over-the-horizon technologies?
I think so. I think Alberta in particular. I won't just say Edmonton. I think Alberta. We're an Alberta entity. We happen to primarily sit in Edmonton, but we have partnerships here. We actually have partnerships globally. We have Ingenuity Lab India, which we've opened up, and we have active partnerships in Israel, in the Middle East, India, Europe, Korea, and other places in Asia. So we have a global presence, but we are the central hub in taking ideas and technologies and weaving them into new solutions for the future.
Right, exactly. That weaving. It seems like Ingenuity Lab is a multifaceted organization today, and it seems like it has very different focuses in healthcare, carbon recapture, agriculture. What is the common thread of all of those?
I think the common thread is the power of N, is where the common thread comes from, and we leverage that to find solution paths that normally are obscure to other investigators, and the reason being is our diversity. We are technically extraordinarily diverse. We have everybody. We have had everybody from social scientists all the way through to gene jockeys and physicists and engineers and chemists. So and physiologists. We have a wide variety of talents that we're able to put together. The second element of diversity that we have is that we hail from 10 different countries. And what that means is that we have people who come all with different perspectives on not only what the problem is, but what an effective solution should look like. And so when you bring this breadth of insight with the breadth of technology, it positions you in a unique position.
Yeah, that is a common theme across every conversation I've had with people at sort of the frontier technologies, is how sharing perspectives seems to be play a key role in developing the further technologies. And as much as I would like to delve into all of the Ingenuity's Labs technologies, I'm going to have to, I think, limit the scope of our conversation to your regenerative healthcare.
Mm-hmm.
Could you explain what that even means?
Well, what we're looking at is can we engineer artificial constructs that replace normal biological function, or can we engage in providing stimulus or to accelerate the repair of damaged organs or tissues or physiological functions, and so this work covers many different dimensions, as you would imagine. Everything from we're developing new ways for designing targeting locales for gene therapy for Duchenne's muscular dystrophy, to regenerative materials for artificial knee meniscuses, to artificial pancreases, and moving on down to some of our exploratory research that's focused on developing a artificial ovary for controlling the differentiation of pluripotent stem cells. So that the idea, if we can control them from the cellular level at the very, very beginning, we may be able to grow new organs directly with no scaffoldings, nothing. Just like a living organism develops and differentiates. If we can control that, maybe that's a path forward. Very speculative. It's one of our way over the horizon type of projects, but it's exciting. And this is all being enabled because we have such a multidisciplinary and very diverse team.
Yeah, absolutely. And could you just tell me, in layman's terms perhaps, what is nanotechnology and how it's being merged with medicine in these ways?
Nanotechnology is just not being small. I know we've all heard the dialogue about how small a nanometer is. And my favorite is a nanometer compared to a meter has an equivalency roughly of a soccer ball to the diameter of the Earth. I mean, so to visually give you the size scale changes that occur. But what nanotechnology really is the precision assembly of matter. It's putting molecules where you want them to be, doing what you want them to do when you want them to do it. Being small is just is just a necessary condition, but it's not sufficient. Otherwise, every time we painted a wall, we're doing nanotechnology, right? So it's putting those molecules and controlling where those molecules position themselves, and then providing a mechanism of controlling what they're doing, so that you can get the function that you want.
So it's really just a tool of getting down to the base building blocks of what our nature, which is atoms and molecules,
Exactly.
and controlling where they're going to be. So could you just give me some examples of what you're working on today, where you can use that idea to create medicine?
I think the artificial pancreas is probably the best. One of the big challenges that people had when trying to transplant islet cells that produced insulin is that ultimately they die because they don't have enough vascularization to give them the nutrients they need, and they can't respond quick enough because there isn't enough vascularization to distribute the insulin when they need it very, very rapidly. So there's the Edmonton protocol, which is the best thing in the world, but it still leaves a lot to be desired. So what we've done is we've developed a material, and what this material does, it has peptide signaling molecules on it. And what these molecules do is they tell the body to do certain things. They talk to the body. So there's one that has VEGF. VEGF is a signaling molecule that tells you to grow veins, and grow a circulatory system. And so we have those in place to tell it to grow. We have others that tell it not to swell, and we have others that prevent it that try and preclude an autoimmune response. And so you have these molecules that are part of the actual polymer. They're not infused in there that dissolve away. They're actually been linked in part of the polymer. So the scaffolding then talks to the body, telling the body what you want it to do. And so we've incorporated islet cells inside these, and we've implanted them in mice. And 20 days later, it's highly vascularized. The islet cells are still producing insulin, and we do this by making this system, and then into a little cone which you use as a needle, and you insert underneath the skin. So it may be they have a way of going into a doctor's office in the future, one day in the future, and you insert it under the skin with the needle, and you cure type 1 diabetes.
Wow.
And it's all done by coupling advances in material science with our knowledge of biology, and then putting in the fact that molecules talk to one another, and putting in those communication pathways and make them as part of the materials that we build and engineer.
Wow, that's amazing. This is quite an open-ended question, so I'll let you take it where you will. But how large of a role do you see nanotechnology playing in healthcare's future, and is it preventative, reactive, or both?
I think it'll be both. I think what you're going to see is healthcare is that we're going to be able to use nanotechnology to short circuit problems that before they become manifest into disease. And we're also going to be able to see that we're going to be able to use nanotechnology to cure or treat disease. So it's a ubiquitous tool. You know, it provides a key that allows us to interface with all of the complexities that go on at the molecular scale in a human body. So it's a key. So what we're learning now is how to file off the right teeth in that key to unlock the right pathways that we want to unlock and control.
Mm-hmm. So we kind of touched on this, but what are the initial focuses maybe of the Ingenuity Lab or of nanotechnology in medicine in general, i.e. what are the lowest hanging fruit? Where are the first things we're
I think there's a number of places with the low hanging fruit. We've talked a little about the artificial pancreas and making biological scaffoldings that are functionally active. There's opportunities, I think, in the computational space of where we use our knowledge of nanotechnology to design new targeting motifs for treating genetic disorders more accurately than we can do now. There's opportunities for establishing diagnostic tools that you would not be able to get access to without having without using nanotechnology as a platform. So I think those three areas you're going to see the most rapid insertion into the medical community.
Mm-hmm. And is it being paired up with the advances we're seeing in gene therapy, delivering Cas9 or the
Oh,
proteins?
absolutely. And that's what I was talking about computationally, is finding new ways of,
Right.
you know, it's there's structure, there's three-dimensional structure associated with biological molecules, and many times the targeting motifs that we use are based on a 2D linear structure. So we say, oh, we have a sequence of genes. What you'll find is that there's a sequence that we normally attribute to for a DNA. You'll know it's the conical base pairing. For RNA, and if you want to insert yourself in the transcription process, they take on a three-dimensional structure, and those conical pairings may not be physically available because they're three-dimensional structures, so it folds over on itself. And so some of the computational work that we're doing is looking at how we can identify where the pieces are exposed, so we can target the exact location, even if they are not immediately available. That's one of the areas that we're exploring. The delivery of agents by having smart particles to deliver the agents in particular locations of the body, are also other areas that I think you're going to find there's a synergy that will develop with some of these other advanced techniques which are just coming online now, like CRISPR.
Right. And those targeted delivery of medicine, I think I saw on your website an example of treating cataracts, for example. I mean, this might be hard to say, but how close is that idea of using the smart particles to go where you want them to go, and how do you program them, if that's the right question?
Well, you program them by having surface activated molecules that again talk to the locations that you want. Say, oh, I want to bind here and not bind there. The trick is not so much targeting the area that you want to, but making sure it only targets the area that you want to. Non-specific interactions are really or is just as big a challenge as targeting, and getting them binding where they don't want them to bind because of other elements associated with the chemistry of the particles that you build. So that's a big challenge, but the ability to target. So, for instance, you talk about our cataract project, and in this case, what we did was we used phage display to do directed evolution on starting off with about 10 to the 9 combinatorial possibilities to identify a RNA peptide sequence that would conform to a specific location within the fibrils that form the cataracts.
Wow.
And so, that takes a lot of work to do that. But when you identify something, it only targets that one area.
Yeah. So 10 to the 9 combinations, are you just basically brute forcing your way through this computationally?
We do this through experiments.
Okay.
And it's a powerful technique called phage display, in which you use a library, about a 10 to the 9 library, and you do multiple rounds of successive refinement to get what's called a consensus sequence that binds to the location that you want.
Do you see the possibility of the Ingenuity Lab being involved in computational representations of biology that are sort of being pursued through artificial intelligence?
We actually have a program here on making a biological computer using ionic currents, by making what amounts to is a really stupid artificial neuron. It has ion channels, potassium, sodium ion channels. It has notchbacks. It has light activated channels as an ATP ACE pump. It gets powered by light. So we have a whole project that we're working on building something like that.
That's really cool stuff. I find that fascinating. Do you think one day it will be possible to represent biological interactions via silicon entirely?
I'm not sure through via silicon, but I think we can definitely do it. Well, eventually in the future, 2400. I don't know.
Sure.
Uh, the the ability in the near term to replace to do metabolic representation on a chip of some sort, I think is very, very, very close. We actually have a technology that we developed here. We call it 4D printing, of which we figured out how to stabilize integral membrane proteins into polymetric structures, and we have a 3D printing system that allows us to put different molecules within a volume that you would want to in space, so you can control the geometry, the structure that you want, and the relative position of different molecules in those positions. So we're in the process now of essentially putting metabolism on in a solid state system.
Wow. If I could delve in a bit further into that, how do you? Because I'm just picturing my crude understanding of 3D printing, where there's basically a nozzle spitting out molecules. Is that the accurate? How do you place a molecule? How do you print a molecule?
We use stereographic light.
Oh, okay.
And what we have is we have polymers that we have been able to, we have that cross-link through using different cross-linking mechanisms and cross-link at different wavelengths of light, and then each one of those polymer monomers gets attached to a biological molecule. So you have a monomer with a biological molecule attached to it that has been stabilized with a unique peptide system that we've developed and patented. So you have a monomer that's biologically functioned. So we expanded the color wheel of physical properties to include biological function in that material. And so then we use light then to cross-link and position polymers with different biological function at different places in space.
I'm going to just shift gears here a bit. On the largest scale, I guess, let's limit it to 50 years. What sort of future just generally do you see when medicine and technology meet? And are you optimistic for the future?
Oh, I'm very optimistic for the future. I think that what you're going to find is that people are going to be living longer, that they're going to live more productive and healthy lives, that many diseases or ailments are going to be rectified. Everything from artificial appendages to the treatment of metabolic diseases are going to become solvable technologically, so that we can dramatically improve the quality of people's lives and life. A bigger question you have is going to be on economic availability, and that's really where the challenge is going to come, because as we all know, the cost of healthcare to the economy is growing at an astronomical rate. Is making sure that everybody has available to the best sources. I am pretty confident, though, that the technology that's being developed is going to drive the cost down. That ultimately we're going to go through a hump of where the costs get higher, and then the costs are going to drop down and become cheaper and more widely available.
Right. So you don't see this sort of dystopian future where only the rich have access to these?
I think it's going to be the opposite. I think what you're going to find is that just like only the rich had cell phones, not everybody has a cell phone. Right, and only the rich people are ever going to have cell phones. But look at what happened with the technology. You go to Africa, and people who make $100 a year, they have a cell phone. Right, and I think that it's the same sort of thing. The costs will go up, and assuming that we have proper governmental policies, the costs will dramatically drop down and become available to everyone.
Right. So once it becomes ubiquitous, the cost will drop. Mm-hmm. While we're on this sort of ethical note, I've been asking some people this question who we brush up against it in our field of biomedicine. If I was to come in here and perhaps condemn you for playing God, how would you respond to that?
I don't play God. What I do is I take inspiration from God, and I use that inspiration to craft solutions. That's what we do. We take our inspiration from nature. We look at the works of the way God works, and then we we look at how we can couple what has been developed within nature, modify it, change it, and allow it to serve a purpose that maybe wasn't originally anticipated.
I like that answer. So with my last few minutes here, just a few questions for me. Do you have any advice for a young person who is hoping to become a budding, I'll call it an academic entrepreneur, such as yourself?
Don't confine yourself to one discipline. The future is having a breadth of knowledge that allows you to be somewhat conversant in many, many different fields. And those fields should not just be restricted to the STEM technology or the STEM squared technology fields. You should be a full person, a person who understands and appreciates fine art and music and has good skills in communicating. And all those things will provide inspiration and perspective and the wherewithal to allow you to be successful.
Right. Once again, it comes back to different perspectives and opening yourself up to that.
You have to do it. If you lock yourself into a room in a very, very focused, narrow area, it's hard to grow. It's hard to see new opportunities because your perspective is very, very you're engaging in tunnel vision. And so what you want to do is open your eyes, open your talents, and move forward so that you can embrace the opportunities as they become available and see the opportunities as they become available.
Right, and be willing to consider new opportunities and don't stay on your path because you think it's the path.
Well, no, don't stay in the path because you think it's the only path.
Right, exactly.
And be open to other paths that are possible. Understand where your goal is and know that in many cases, there's more than one path to get to that goal.
Right, and often there's going to be obstacles in that path, but the obstacle actually ends up becoming the path itself.
Yes.
You can pivot. I thank you so much for your time, Carlo.
It was my pleasure, and best of luck on your studies.
I appreciate it.
And I'm sure you can do great.
Okay, thank you.